My name is Misha Ahrens. I also work at Janelia. I work a lot with Jeremy. And I'm going to start off with a terrible joke describing what we study. And I'm making this joke because it's the theme of this meeting. We study the brain, and to do this, we use fish and chips. I promise no more jokes for the rest of the talk. Instead, I'm going to talk about two types of technology that we've developed in order to do what we do, and then a computational framework that Jeremy's developed in collaboration with us. And finally, if there's time, I hope to tell you about some biology that we've done with this. So we studied the same animal, the larval zebrafish that Michael just talked about. We use a slightly different imaging technology. Instead of light field microscopy, we use light sheet microscopy. This is how it works. So just to compare to another microscope that's very common that Nick talked about as well, which is a two-photon microscope. This images a volume, so it can register neural activity in a volume by scanning around a single point of excitation. So this is a great technique, but it's slow for three-dimensional volumes because you have to scan one point in 3 dimensions. With light sheet microscopy, instead of using a point, you use a laser beam, basically. And to do this, to scan a laser beam around, you use a second objective. And literally, the way you take a volume is to do what I'm now doing with the laser beam, with my laser pointer. You scan from the top to the bottom. It's slower than light field microscopy, but has a higher spatial resolution. But you'll see that the data we get out is comparable in its nature. Here's a drawing of what this looks like in reality. This is the earlier microscope we used. We've now redesigned it to be able to do this in combination with behavior. Now, on this slide, you can see a larval zebrafish brain. This is the orientation. This is about 0.5 millimeter long and 1/4 millimeter deep. And this over here, the eyes, the spinal cord starts here. You can see lots of cell bodies, and this is about the speed at which we're imaging. We're going a little bit faster right now. And one example data set of what comes out is this. Oops. So here the brain is sliced into 40 slices. They're spaced close enough together to capture all the cell bodies within the volume that we're imaging. And in gray is the anatomy, and in color is the neural activity superimposed on the anatomy. This is a fish that's spontaneously, whose activity is spontaneously evolving. There's no stimulus, and we don't have a behavioral readout. You can see that, nevertheless, the brain is highly active. And so to summarize, we get about 80,000 neurons out of about 100,000 at a couple of hertz in single-cell resolution. Here's a top side and a front projection of this data. You can see the single neurons more easily. And this data is complex, and we've been working with Jeremy Freeman to create analysis tools also running on Spark for analyzing these data. And here's an example of brain volume that comes out of it. This is a volume that describes direction tuning. Here we used a moving visual stimulus that's moving in different directions. And every cell is most sensitive to one of these directions or doesn't care about the direction. Or is not responsive. So every color here represents one direction to which the cell is most sensitive. You can see that there are lots of cells in the brain that are direction sensitive. This both illustrates what we can do with this technology is quickly analyze these data, but it also represents the challenges that we're facing, because if two cells are tuned to the same direction, they might actually be extremely different in their nature. For example, a visually tuned cell over here might simply represent the visual environment, whereas a directionally tuned cell here in the hindbrain might actually represent behavior. And other cells might represent combinations of these or a dynamical interaction between these variables. So understanding this data set, even after doing this initial processing, is extremely challenging. Now, we wanted to not only do imaging of a passive animal, but we actually wanted to have behaving animals in order to really make sense of the role of the brain in behavior. So now we come to the second piece of technology. Here we developed a way to image the brains of behaving animals, or fictively behaving animals. So here's an animal doing a freely swimming behavior, it's just swimming in a dish. What they do in response to whole field motion is they swim along with it. This is called the optomotor response. This is this behavior recapitulated in a paralyzed animal. The way this works is we paralyze the animal, we record electrical activity in the tail coming from the spinal cord projecting to the muscles. The muscles don't respond to this activity because the animal is paralyzed, but the spinal cord in the brain is still completely functional. So we have an electrical correlate of behavior which we interpret as intended behavior, and we can now use this to drive virtual locomotion through a virtual reality environment. And that's exactly what's happening here. So this fish is playing a computer game that's recapitulating exactly this behavior, but now it's paralyzed. All right, so to combine the light sheet technology with this fictive behavior technology, we built a new microscope with a chamber. The fish is sitting here on a little pedestal. We're recording electrical activity from the tail, and now we use two laser beams, one from the side and one from the front. The reason for this is because we don't want to shine the laser into the eye, but we still want to get the entire brain. So the side laser scans from the side whatever it can, but whenever it's positioned over the eye, it switches off very quickly. The front laser records everything that's between the eyes. And so the data set I showed you earlier was actually recorded using this system. The question now was whether the fish would behave naturally in this situation, because it is a little bit, there's stuff going on. There's an objective moving, there's some blue scattered laser light. So we tested the optomotor response that I just showed to you. When the stimulus is on, the fish swims. When the stimulus is off, the fish doesn't swim. And it doesn't matter to the fish whether we're scanning the brain here or whether we're not scanning the brain. So this is promising. We can image the entire brain during behavior. And here's an example of this. This is the stimulus here. It's again a moving grating. When the grating moves, the fish swims along with it. This is now the strength of swimming, this dot. And here's whole brain activity. It's a projection from above. And again, so when the fish moves, there's a lot of activity in the hindbrain. In the forebrain, there's always activity, whether the fish is swimming or not. So these brain areas, different brain areas have very different properties, and we're trying to understand how they interact. All right, the third piece of technology is computation. And this is all collaboration with Jeremy. He built a library of analyses on top of the Spark framework for distributed computing, including dimensionality reduction, univariate analyses, including regression, and so on. And the idea is to expand this library to include more complex analyses. He called this Thunder, and it's freely available. So now that I've covered the three core technologies that we use, I want to talk a little bit about how we actually use it for biology. So this is one instance of something we've done in the lab. So putting it all together, behavior, whole brain imaging, computation, and anatomy. The topic here is summarized by the following title: Neural Control of Spatiotemporal Patterning of Spontaneous Locomotion. This is also in collaboration with Tim Dunn and Eva Naumann, whom I hope have now arrived. Yeah, there they are. Hey. So the idea here is that animals respond to stimuli, and they can solve problems. But when they're not faced with any specific stimulus or any specific problem to solve, they still show very rich behavior. And a lot of what we do is actually self-generated behavior in that sense. So years ago, we were interested in how zebrafish behave when they are not challenged in any way. So Tim did this experiment where he filmed a zebrafish that was simply swimming in a completely featureless environment. Equiluminant light, no olfaction, no visual stimulus. And the fish swims around, turns left, right, swims forward. And this is real time. So you can see that the fish swims in discrete swim bouts. And every swim bout has this property, an angle and a forward component. Now, at first sight, this looks random, but if you look carefully over time, if you color the turning direction magenta and green, depending on whether he turns left or right, you can see that over time, there's actually very interesting structure to this behavior. Namely, he strings together, turns to the right, and turns to the left on timescales of about 10 seconds. It's still stochastic, but there's a very strong statistical structure. And this is especially clear if you look at the projection of this path. You can see these strings of magenta and strings of green. And if you rotate the fish and look at the future trajectory, depending on whether he just turned left or right, you can see that for about 10 turns, the directionality is quite consistent. So we were wondering for a while where this might come from, and we thought maybe in the forebrain, because in the forebrain there were similarly slow timescales as in the behavior. We imaged there a little bit, nothing came out. Then we realized that if we have whole brain activity in hand, simply by covering much more of the brain than we could before, we have a much better chance of finding cells that might drive this behavior. So we recapitulated the behavior in this fictive setup in which the animals are paralyzed. We tested whether it's present. Now, in this fictive setup, the animals are paralyzed, but we're recording the behavioral, the electrical correlates of behavior, and we can actually decode whether the fish wants to turn left or right or swim forward, and by how much. And so if we decode the behavior in this way, we can reconstruct the fictive path through a virtual environment. And what we see is that it looks very similar to the path of the freely swimming fish. So qualitatively, these are extremely similar. These slow timescales and unidirectional behavior are present in both. Now, given that we can do this in paralyzed fish, we can now port it to the light sheet microscope. And this is what the data looks like. Again, this is a top projection. Right turns are shown here, left turns here, and this is whole brain data shown as a projection from above. So we then analyze this by assigning to every voxel, there are a few 100 million voxels in this data set. Every voxel got assigned a behavioral tuning described by a behavioral tuning space. The fictive swim signals were decomposed into an amplitude and a direction. The amplitude describes how strong the fish swam. The direction describes whether he wanted to swim left or right. And so at any point in time, the fish sits in one point of the behavioral tuning space. We can then turn this into 12 regressors, regress whole brain activity against this, and describe every voxel according to one of these sort of behavioral receptive fields, which are analogous to sensory receptive fields. Now, here are some volumes that come out. This is the volume corresponding to a forward-tuned behavioral component. So these neurons and pieces of neural pale active when the fish swims forward. And this volume describes which pieces of the data are active when the fish is turning left or right in green and magenta. And just from this very simple analysis, in a sense, we have this two-dimensional behavior, the left and the right side of the tail. From simply doing a regression based on these two variables, we get this very clear anatomical structure. You can see single neurons. You can see neurite tracts. You can even see these two, what look like two single neurons in the forebrain. So very clear anatomical structure from this very simple analysis in about 20 minutes of data. So this gives us great hope that we'll be able to use these technologies for solving more complex integrative problems in the future. Now, these patterns are consistent across fish. Here are 4 of them. All of them show this structure in the hindbrain here, which we called the hindbrain oscillator, because for lack of a better name, we don't know yet in mammals what it corresponds to. And if you align multiple fish, then you can see that the anatomy lies very closely, is very closely aligned, which is great news because this means that we can compare these computationally derived volumes to anatomical volumes, anatomical atlases of cell type, brain areas, and gene expression, and really go from function to anatomy and combine the two and use those to go forward for more specific studies. So what I've just shown you is a relationship between this functional activity in the hindbrain oscillator, also other areas, but in particular in this area and this spontaneous behavior and the slow statistical structure in in this behavior. We followed up now with more specific experiments in which we ablated one side of this structure and left the other side intact. And what happened was that after the ablation, the fish would swim a lot more in the direction of the intact side, showing that there is a causal relationship between these sets of neurons and behavior. So to conclude, just to summarize this, I skipped over a couple of slides that would explain this in more detail, but we found this 4 clusters of neurons in the hindbrain, which we think implement a two-state model or two-state system that makes the fish turn left or right with long temporal correlations, which eventually we believe might implement an efficient foraging strategy when the animal has no guidance cues from the environment, at least no explicit guidance cues from the environment, and has to rely on internally generated signals to guide its behavior. So a lot of this work was done by Timothy Dunn, Yumu, Sujata, and Jeremy. Here's a recent photograph of actually 3 labs at Janelia. And here are more acknowledgements. As I said, Tim, Don, Yumu, Jeremy, also Eva were involved in the latter project that I described. Nikita built the microscope, and we're indebted to a lot of other collaborators and supported by Howard Hughes. Thank you.